Yes, good evening, everyone, once again, and welcome back to our Neurotorium peer-led training series on depression this week. So yesterday, Dr. Katama took us through a brilliant exploration of the epidemiology and diagnostic formulation of depression, and in the process, he laid a clear foundation of how we identify and frame this condition within our clinical work. And Dr. Katama, your presentation was engaging, practical, and it gave us a lot to reflect on. So today, we are moving a step deeper, and that is into what I like to call the real meat of the discussion, and that is the etiology, neurobiology, and the cause of depression. So this, ladies and gentlemen, is actually where science is meeting meaning, where we are going to begin to understand why depression happens, how it unfolds in the brain, and anyway, what pathways it uses, or what pathways shape its course over time. So we are once again privileged to have Dr. Sadat Katama. This is a quick reminder that for each week we are going to be handling one topic. So for this week, we are focusing on depression. Yesterday, we had the introductory information given to us. Today, we are having the meat, and tomorrow we shall have the icing on the cake, if for lack of a better word. So stay with us today, tomorrow, and the other reminder is for each of these days, we are actually going to be using the same link. So there is no need for you to generate a new link. Just save that link, and we shall be using it throughout the series. So colleagues, let us settle in and stay engaged. Dr. Katama, if you're online, I request you to take the floor. Take us away. Thank you.
Yes, thank you, Dr. Mianga. I hope I'm audible.
You are very audible.
Yes, good evening, everyone. Welcome to the second day of our sessions on depression for this week. And like Dr. Mianga has said, throughout for the next 6 weeks, we shall be discussing one topic at a time. And for this week, the first week, we are looking at depression. Yesterday, we went through the definition of depression, how to diagnose depression, how to rate the severity of depression. And today, we would like to look at the real neuroscience behind depression. And in this, we are going to look at the etiology and the neurobiology. Particularly, our attention will be given mostly to the neurobiology of depression. I'm Sadat Katama, and let's take it off. So, when we talk about the etiology of depression, it is not a one-factor point that this is one thing that causes depression. However, there is usually an interplay between different factors to lead to bring about the symptoms that we talked about of depression yesterday. We find genetics as one of the genetics as one of the factors, and then we find the environment, which plays a significant role. This one comes at different points. It can be in that time when the individual is actually is experiencing this depression, or it could have been way back in duration, that it happened way back, like the childhood maltreatment, and then of course there can be chronic stress. So all of these can predispose to depression. Then we have the biological and then the psychological factors, of which genetics, well, it is also biological, but here we are looking at the actual pathophysiological pathways, the inflammatory response, the monoamine pathways. And then for the psychological factors, things like the cognitive patterns of an individual can all drive about depression. Now, depression has been studied for quite some time, and not only humans do get depression, but even some even the other animals, lower animals, also do experience depression. Now, when you look at this study, this is we have the first swim test where a mouse is dropped in water and then observed for how it attempts to survive. You find that if these mice struggle to keep afloat, then this is a sign of antidepressant response. Basically, it is fighting for survival. When you look at the tail suppression test, similar thing is happening. When you suspend the mice by their tails, they are basically supposed to keep fighting to try and get off from that. And that is a sign of antidepressant effect. Basically, it is fighting for survival. While if you find it just lying there without any particular response, you know that it is actually failing to fight. Now, most of the talk is going to involve some really complex mechanisms. We shall be talking about things like stress, exposure to trauma. We shall talk about the HPA axis. We shall talk about inflammation. However, we want to start with the genes and then the environment. There is something we call epigenetics. Basically, the interaction of the environment and the genetic makeup of the individual, which then leads to the switching on or off of the genes, of the transcription factors of the genes, which are necessary in the modification of particular enzymes, for example. Then this can lead to different activation of proteins, DNA, and these can lead to either the manifestation of the symptoms we see or even alleviation of those symptoms. So we are going to talk about epigenetics quite for quite a moment, and then we shall go ahead and talk about the HPA axis, the role of the glucocorticoids. We talk about the activation of transcription factors and how these trigger inflammation, how these trigger depression. And of course, then later on, we shall look at the mechanisms of action of the different medications that we use in the treatment, what they specifically do target. Now, the pathological hypotheses basically and the mechanisms behind depression are quite many. We shall be talking at one at a time, and the first one is the genetics of depression. Now, we have what we call the genome-wide association studies. These have been done and they have provided significant insight into the mechanisms underlying depression and, of course, the genetic risk factors. You find that studies have shown that children of people with depression have a 35 to 40% chance or risk of developing depression themselves. This one is roughly double what we would call the risk in the general population. So general population is for people who do not have parents with depression. So you find that having a parent with depression increases the risk by 35 to 40%. And when you look at twin studies, there is a heritability of about 37% for depression. However, you realize that this is actually the lowest than most of other mental disorders. When you look at things like schizophrenia, the heritability is way higher for the twin studies, about 60%. So for depression, it is quite lower for compared to other mental disorders. Here, this diagram looks at the interplay between the environment, weather, chemical exposure, stressful life events. All of these are going to affect these elements here and then the different pathways. But ultimately, the impact is going to be seen at the synaptic junction, at the synaptic function, because this is the functional unit of the brain, basically. So this is where we are going to find the actual change that is going to lead to the development of depression at this point. And we shall be talking deeper about this. Now, what genes are involved in depression? Several genes have been implicated. Of course, no one single gene has been found to show that its presence it automatically leads to depression, but the interplay between these genes and the environment and other factors can then potentiate the development of depression. You find that this table here looks at the gene, then looks at the known functions and the roles of those genes, and then the representative impacted tissue in the brain. So when you look at the NEGARO one, basically it is to do with the brain volume, especially the hippocampus, involved in social behavior and non-social interest. So depressive and anxiety-like behavior, these are some of its functions. And so you realize that if you have issues within this gene, then you're going to have issues along those lines. When you look at the DRD2 reward depressive-like behavior, the nucleus accumbens is very important in the reward system. So if you have something wrong with this DRD2, then you're going to have an impact around the nucleus accumbens. And then someone whose motivation is going to be low, someone will lose kind of the satisfaction they would get in certain either activities or things that probably they used to enjoy. So the genes play a significant role, and we need to always pay attention. That's why when we are taking the history, we always ask about, do you have a family history of depression? So that we try to link a potential genetic predisposition. Then we look at the environment. Usually in the environment, it poses both risk and protective factors. So this flow here shows the risk factors on the left and the protective factors on the right. So we're going to look at all of these things. When you look at the social determinants of health, when you talk about the social determinants of mental health, you're going to find things like the demographic factors. Someone being a female puts them at a higher risk of developing depression compared to a male. Then, of course, adolescent and younger adult age, low socioeconomic status. When you look at things like poverty, unemployment, these are very, very strong points in as far as depression onset is concerned. Meanwhile, when you look at the protective side, economic security, social protection, when you look at healthy lifestyles, physical activity, we are going to look at the neural pathways and where some of these things come in. Day-night structures, basically all of these things are kind of protective against depression. So usually the goal is to ensure that all of these environmental risks are reduced while improving these protective factors as far as the environment is concerned when we are managing our patients. Studies have been done, and we look at the childhood maltreatment, for example. It has been shown that individuals who had a rough childhood, retrospectively, their odds of developing depression are 2.21, while prospectively it is 1.56. In both cases, whether you follow up from a point towards the future or you go back in time, you find that the risk of developing depression increases with the presence of childhood maltreatment. A study in China looked at air pollution, and it concluded that increase in certain emissions, daily emissions, were associated with increased hospitalization for depression. As you can see, the citations are there within the literature, and of course, when you access the slides from the Neurotorium website, you can actually go deeper to look into these studies and what exactly they did and what exactly they found. Of course, there are these other risk factors. Now, when we are looking at the risk for depression, we use what we call the biopsychosocial model. Most of the part that I've been talking about is genetics, environment. However, we have now what we call the biological factors that drive depression. These biological factors are those inherent things within the body. You find there are those which are purely central processes only happening within the brain, while there are those which are central and peripheral, cross-cutting between the brain and the peripheral system. So you find things like the neurotransmitter system. Here you look at the neurotransmitters like serotonin, noradrenaline, dopamine, GABA, glutamate, as we shall be seeing later on. Then the brain structures and function. We shall look at the structure and then the function in terms of the neural pathways. Then neuroplasticity, the change within the brain connections that happen. Basically, these are purely central. While those that combine central and peripheral, we have the immune stroke inflammatory system. These systems might be within the blood or might start from the CNS, but then lead to a cascade of events that can affect the entire body. So we shall look at the peripheral and central expression of immune factors, the role of the microglia and the astroglia, and then the kynurenine pathway dysregulation. Then we have, of course, the gut microbiota brain axis. This is one of the newer models explanations for onset of depression. Then we have the HPA axis, the hypothalamus pituitary adrenal axis. This one related to stress, and we shall see in detail how it can lead to depression. Then the epigenetics, which we've already talked about, the genetics and the environment playing together to lead to depression. This diagram here, this graph looks at the risk of suicidal behavior. At this point where you have these protective factors, things like social support, maybe someone is on medications, you find that the risk of suicide is actually low. However, as you go along and you have changes in the serotonin system, you have impulsivity setting in as one of the symptoms, poor decision making, then you have aggression, you find that then the risk of suicide tends to increase. And this suicide risk goes ahead to reach a point where the individual then starts to attempt suicide. However, when it becomes too much, you find these harmful effects, then individuals can actually go ahead and complete suicide. So we have different factors that can modulate suicide in depression. We have increased levels of inflammatory mediators that we shall look at. You may find molecules that are produced by neurotropic pathogens like toxoplasma within the brain. Then, of course, lower levels of testosterone can actually lead to increased risk of suicide. And then, of course, the dysregulation within the neural pathways, inflammatory cytokines, the kynurenine pathway can all increase the risk of depression. So these we are looking at so that as we are seeing these patients, we try to think through whatever is going on within the patient so we understand how best to improve. Where you find management is not working, you should think through all lines to be sure that you are targeting all these possible pathways, possibly. Now, let's talk about the neurotransmitter systems and depression. So for the neurotransmitter systems, these are the different neurotransmitters usually that can be implicated. These are very important within the body. So you find serotonin, which has the receptors, the 5-HT receptors from 1 to 7 and their subtypes, noradrenaline, dopamine, then glutamate, the major excitatory neurotransmitter of the brain, and then, of course, we have GABA, which is the major inhibitory neurotransmitter of the brain. Then we have acetylcholine and histamine. All of these neurotransmitters play a significant role in modulation of the activity of the brain. You find that these do not work in independence. For example, where serotonin might be excitatory, GABA might actually be working to inhibit that neuron. On the same neuron, you may find receptors from all these neurotransmitter. So depending on which neurotransmitter is released or is in high concentration at that time, then you'll have the effects of that particular neurotransmitter. So these we look at these so that we can understand our targets when we are managing the patients. So what happens within these neurotransmitters to lead to either depression or alleviation of the symptoms? Now, most of the symptoms of depression basically implicates several brain structures and regions and different neurotransmitters. You look at the functionality of one neurotransmitter being able to affect another. For example, where serotonin's function increases, you may find that actually the activity of another monoamine like dopamine or noradrenaline actually decreases. So the major three monoamine systems that are involved within the pathophysiology of depression are serotonin, noradrenaline, and dopamine. And this, what we are looking at as the monoamine hypothesis, basically says that a low or abnormal activity of these neurotransmitters is what is responsible for the corresponding features of depression. So you'll find that if someone has low levels of dopamine, then motivation is low. So they don't find pleasure in things they used to do. While if serotonin is the one that is low, then you'll find that you're having more of the low mood and then the cognitive, or the low mood that will be happening within this individual. So it is important to understand which system you're working with, which symptoms are existing in the patient at that particular point in time, so you know your target. This diagram, I'll take some time on this because this is where we are going to see the effect of our treatments that we usually give. So what you're looking at here is the synapse. So you have this up here is the presynaptic membrane, and this side is the postsynaptic membrane. In between here is the synaptic cleft. So on the presynaptic membrane, usually we have the presynaptic knob, this side where you have the cytosol, and in here we have where serotonin is synthesized, stored, and broken down, from which it is released to the cleft. What you're seeing here is the serotonin transporter. The role of this protein is basically to take serotonin out of the cleft back into the presynaptic knob where it is stored or broken down. On the postsynaptic membrane, you're looking at, this is the serotonin receptor right here. So when serotonin is released, basically it is supposed to attach to this to lead to effects in the postsynaptic membrane. At this point, the serotonin transporter is functioning normally, and you realize that serotonin is being pushed back out. And there are low levels of serotonin within the cleft, which means binding to the receptor would also be less. And this is what happens in depression. In the presynaptic knob, you find the monoamine oxidase enzyme. The role of this enzyme is to break down the serotonin. When you break down this serotonin, it is not going to be released back immediately into the cleft. So you're going to have high activity of the monoamine oxidase enzyme and then the activity of this serotonin transporter, all ensuring that less serotonin is within the cleft and less of it binds to the receptors. So this is what leads to depression. Our medications are going to target either this serotonin transporter or this enzyme to ensure that we have an increased concentration of serotonin within the synaptic cleft. The higher the concentration of this neurotransmitter, the higher the chances of binding to the receptors on the postsynaptic membrane and then affecting action potentials within the postsynaptic neuron. So medications like the serotonin, noradrenaline, selective serotonin reuptake inhibitors, the serotonin noradrenaline reuptake inhibitors, or the tricyclic antidepressants, they all inhibit this serotonin transporter. So when you block this serotonin transporter, the serotonin within the cleft will not be pumped back into the presynaptic knob. So you'll have increased concentrations of serotonin within the cleft, and then, of course, transmission. If you don't use that, then you can use what we call a monoamine oxidase inhibitor, the myo-inhibitor. This drug binds to the monoamine oxidase enzyme, inhibiting the breakdown of serotonin. So you'll have more serotonin within the presynaptic knob, which will then be released into the synaptic cleft. So you'll have higher concentrations of serotonin within the synaptic cleft, which will bind to the receptor on the postsynaptic membrane. So basically, this is where most of the medications that are used within management of depression target. They target the serotonin transporter, of which you have the TCA, the SNRI, the SSRI, while the monoamine oxidase inhibitor targets the monoamine oxidase enzyme, increasing concentrations of serotonin. I have not mentioned examples of these medications, the SSRIs, SNRIs, TCAs, the MAO inhibitors, but of course we shall find these when we are looking at the treatment principles tomorrow. So this is one of the most important mechanisms that explain the onset of depression, but as well as the management of depression. So when you're treating your patient, you should be thinking along all these lines. Then we look at glutamate and GABA. The role of these neurotransmitters is just coming up, and there is newer evidence that actually these neurotransmitters might be playing a very important role in the onset of depression compared to how it used to be believed. For example, you find that glutamate is basically the major excitatory neurotransmitter within the brain. However, glutamatergic abnormalities have been observed in the CSF, in the plasma, and in the brain tissue of people with mood disorders, including depression. While it might not be easy to measure some of these neurotransmitters in living individuals, for example, for GABA, there is evidence that actually dysfunctions within these systems might be contributing to depression. So you find studies that have shown chronic stress affecting GABA and glutamate pathways. Then these then lead to reductions in the volumes of the prefrontal cortex, the hippocampus, and yet we know that these structures are involved in particular elements of functions within the brain and the symptoms that we see in depression. So a drug called ketamine, which is used as which is quite common to us as an anesthetic, it is a glutamate antagonist. However, it has been shown that its effects can actually have dramatic effects on depression within hours compared to the weeks that we see with the monoamine antidepressants that we've been describing. So this underscores the role of this system, the glutamate system, when it comes to the pathophysiology of depression. You find that when you use ketamine, depression actually improves within a very short, the symptoms improve within a very short time. Now, this clearly shows us that we need to always think through about what exactly is going on. Yesterday, someone asked about treatment-resistant depression, neural pathways that could be possibly explaining. So where you're giving someone an antidepressant, a monoamine oxidase inhibitor, or you're giving an SSRI, you may find that those medications are not working, or the patient is not responding, and you say, this is treatment-resistant depression. But when actually the defect is in the glutamate system, of which if you give ketamine, you find that actually the symptoms alleviate. And indeed, ketamine is used in the management of treatment-resistant depression. Now, what brain changes do occur in depression? I mentioned about neuroplasticity. Basically, neuroplasticity is the ability of the brain to change its activity. This comes in response to either intrinsic or extrinsic stimuli. So there is reorganization of the structure, the function, or the connections. If you hear something or you read about something, a new connection is formed between different neurons to allow for that information to be processed and stored. So this is neuroplasticity. We have neurons and glial cells within the brain, which come from the stem cells in this process called neurogenesis. These are the ones that form the structures of the brain. Once these neurons are formed, that is when neuroplasticity takes place. Now, studies have linked certain elements of neuroplasticity to depression. You find that if you have reduced levels of the growth factors like the brain-derived neurotrophic factor, BDNF, it increases the chances of depression. So when you look, the study looked at individuals with reduced BDNF and NGF in people with depression compared with controls, you find that there is a difference in these growth factors in individuals with depression versus those without depression. And of course, then the use of certain medications or certain activities that can actually improve or increase the concentrations of these growth factors, things like physical exercise, have been shown to improve the symptoms of depression in some individuals. Whenever there is a change within the brain, it is important to study this change and in which part of the brain it is occurring. So you look at the study of the brain function, and we can do this using the molecular imaging. So when you look at the MRI spectroscopy, the PET scans, they are basically going to look at the concentrations of the different receptors within the brain. Basically, this one looks at the different receptors within the brain, and the different colors are going to show different concentrations. Now, this one basically is for the serotonin 5-HT2A receptor. So the blue is at 15, while the very red is at 60. So it is important to sometimes study this so that you know whether the problem is with the serotonin receptors or it is something else that you're dealing with. So for places where it is actually possible to do these studies, it is really imperative that you follow through with such studies. Of course, in our setting, you find that we do not have some of these modalities, so it really becomes hard to do these studies. However, a study looked at 87 patients with depression, and they were studying they wanted to quantify the hippocampal 5-HT4 receptor binding within with exposure to a selective serotonin reuptake inhibitor. And you found no significant change in the mean hippocampal volume over the course of 8 weeks of SSRI treatment was observed. However, the 5-HT4 receptor binding was negatively associated with the hippocampal volume within women. That means that sex-specific link between serotonergic signaling and hippocampal plasticity may not actually be sufficient. So there's insufficient evidence as far as that is concerned. So you are not going to say that because depression occurs more in women, then the SSRI will work more in women than in men. This study basically looked at that, and it showed that there is not really particular sex-specific difference within the response to serotonergic signaling and hippocampal neuroplasticity. So when we talk about this neuroplasticity, basically we are looking at the neurostructural alterations that happen within when in depression. So structures like the prefrontal cortex, the anterior cingulate can be affected in depression, and yet these are important in regulating emotion, attention, cognitive control. And a study looked at adolescents with depression, comparing it with those without depression. And it found that the brains of adolescents with depression had a lower total surface area. This clearly tells you that depression actually shows that depression coming up can come from a global reduction within the brain size, the surface area of the brain. And this is seen more strongly within recurrent depression. Yesterday, we talked about these kinds of depression. So the more recurrent the depression is, you realize that the more damage there has been to the brain structures. So it is important sometimes to do these studies. You do MRIs so that you know what you're dealing with for a particular individual that you're seeing. So you can find white matter microstructure abnormalities that then that would point to disconnectivity between the different systems. And since we say that these systems work in unison, this can then lead to the manifestation of the symptoms that we see in depression. Then, of course, we have the neurofunctional alterations. Those particular functional within the network, the changes that happen within the connections leading to onset of depression. So you find that individuals with depression usually spend significantly longer states characterized by weak functional connectivity within all brain networks. This is, of course, proportional to the time spent with the symptoms and also the temporal stability, and as well as the severity of the depression. So it is important that we think through all this when we are thinking depression. Then, of course, we have inflammation. The immune system basically plays a very important role in modulating depression. You find that various factors, we look at the pro-inflammatory cytokines being involved in the onset of depression. So you find that this side we have in the energy saving intake response. You're going to find some of these as symptoms of depression. However, this then do modulate the immunometabolic stress, which then can lead to activation of these, for example, interleukin-6, tumor necrosis factor alpha, these adipokines, the hormones like insulin, which then will lead to this altered neural connectivity. This is very important because sometimes patients will come and you're giving you SSRI, it is not working because you have not looked through the potential of inflammation being the cause. So the immunomodulators, like the cytokines, have been shown to, of course, these within the brain, you have the microglia that are basically the immune cells within the brain, now within the CNS. So when you have inflammation, you're going to have increased activity of the microglia. At the same time, you're having a reduced activity of the astrocytes. This then modulates the integrity of the blood-brain barrier. Then there is recruitment of the monocytes, which will then, because you've compromised the integrity of the BBB, then these monocytes from the peripheral system can actually join the CNS, and then you're going to have damages within the neural systems in the brain. So the tumor necrosis factor alpha, some therapies are being developed targeting these cytokines. The NSAIDs have been used in some studies, but of course with inconsistent results. Anti-inflammatory antibiotics have all been used, but with inconclusive results. Then we look at stress. Stress is a very important risk factor for the development of depression. Basically, the hypothalamus pituitary adrenal axis appears crucial within the development of the symptoms of depression. Patients with depression usually have high circulating levels of cortisol from the adrenal glands. Then these this high levels of cortisol is going to modulate. Of course, it will definitely, it is supposed to have negative feedback on the hypothalamus, the pituitary, as far as release of the STH is concerned. Now, evidence has shown that individuals with, for example, Cushing's, where you have very high levels of glucocorticoids like cortisol, they actually tend to exhibit depressive symptoms. And when you manage the Cushing's, the cortisol levels, the depressive symptoms also alleviate. So the long-term effect of stress on the HPA axis leads to an overactivity of this HPA axis. When there is overactivity of the HPA axis, then there will be neurotoxic effects on the hippocampus. Then you'll have reduced volume of the hippocampus. At the same time, the effects of cortisol can lower the BDNF levels, which then also leads to reduced hippocampal volume. Then, of course, stress still leads to depletion of serotonin, noradrenaline, and dopamine. So all of these play together within this HPA axis to lead to the onset of depression. This is still what we've been talking about in stress and depression, basically the HPA axis. So you're going to have issues within the neuroplasticity, neurogenesis. You have the adrenal glands releasing too much of the glucocorticoids, cortisol. This is going to lead to increased levels of, for example, inflammation, oxidative stress, of course, and then you can find things like mitochondrial damage and dysfunction, as well as the increased glutamate neurotransmission. These, when you combine them, they are going to lead to structural changes and functional alteration within the brain, which is then going to lead to depression. Finally, we have depression and the gut. This is where newer research is actually coming up, showing that there are possible approaches impacting the gut microbiota that are that modulate depression. So the presence of prebiotics, those are the non-digestive fibers, which are usually digested in the GIT, and then they support the growth of helpful bacteria, as well as the probiotics. Now, the probiotics are the actual gut bacteria that are helpful. These ones can modulate the release of certain inflammatory markers. So you find that the bacteria within the gut can modulate inflammatory pathways, which can then be implicated in depression. So for individuals, when you compare individuals with depression with those who do not have depression within the population, you'll find higher levels of allistips and the parabacteroid species in persons with depression, while they will also have lower levels of the Prevotella species. While this is known to be a particular factor contributing to the onset of depression or modulating the symptomatology within depression, the therapeutic treatments that are basically targeting this are still just under development and are being researched on. So probably in the future, we shall have agents that will be targeting this gut microbiota, so the gut-brain axis, basically, all leading to depression. And you're going to find that there are going to be these other things playing along: the stress, HPA axis, the neurotransmitters, the immune system, the cytokines. Then, of course, you're going to find things like nutrition, the different components within the food that individuals actually eat. You're going to find, because these neurotransmitters are actually coming from amino acids, which are supposed to come from the diet that individuals are eating. So you find that when you're having dysfunctions or poor nutrition, it can also predispose to this depression. In summary, we've looked at the neurobiology of depression. And yesterday we said depression is a complex and heterogeneous disorder, usually driven by a range of factors, including genetic, environmental, biological, and psychological factors. The genome-wide studies are giving us more relevance to the heritability of depression, and much focus is, of course, on the biological factors, which we can modulate with the medications, the HPA axis, the immune and inflammatory systems, the gut microbiota, the structure and function alterations, including the monoamine system. Then, of course, the behavioral, psychological, and social culture factors also play a role. And where you have high socioeconomic status, good parenting practices, social support, and all the other protective factors actually are helpful in reducing the risk of depression. Thank you very much.
Wow, wow. Dr. Katama, thank you so much for the fantastic session. The topic of the neurobiology of depression is a real heavyweight, and it is a swamp which, if you are not careful, you can easily get lost in. But you've somehow managed to actually trim it down into bite-sized portions that we could all swallow without choking, if I may say that. So we truly appreciate the clarity and patience that you have brought to this fairly complex subject. So, colleagues, it is very important to note that what Dr. Katama has shared, if you've read through the slides, is actually a very small portion. So we still encourage you to go to neurotorium.org, log in, join, sign up for their newsletter, and download most of the content that they have for you to better digest what our colleague was talking about. I would also like to remind us that every Tuesday, Wednesday, and Thursday for the next 6 weeks, we are going to have a different topic. An example is next week, we are actually going to be talking about substance use disorders on Tuesday, Wednesday, and Thursday at 7 p.m. And we've decided that we are not going to follow the mantra of African Meridian time. We shall be starting at 7 and ending at 8 exactly. Allow me to appreciate all of you for making it here, and most of all to thank a number of seniors I can see within the chat who as well listening in. I can see Dr. Emmanuel Pamizo. I'm going to request you to unmute and quickly say hi to us before we delve into the session of question and answers. So I'm requesting each and every one of us to write um, what would like to ask within the chat. Dr. Pamizo, I think I've given you permission to unmute, so please just say hi to us.
Yes, thank you very much. Thank you, Dr. Muyanga, for that information. Thank you, Dr. Katama, for your presentation. You've highlighted very well where depression comes from. People sometimes think that psychiatry is just a matter of forgery, but you've made it clear that psychiatry is based in biology. People don't just forge. It's not an issue of witchcraft. It's not an issue of people having weak minds. They are actually biological reasons which can be measured and which can explain why depression specifically comes into existence. I would like to thank all the medical students who are here, specifically the ones from Guru University. I had asked them to add the word GUM at the end so that I can easily identify them and, keep track of them. But I know we have students from all over the country, Mbarara, Makerere, Soroti, all over the place. So again, thank you very much, Dr. Muyanga. Thank you, Dr. Katama, and thank you for the organizers. We are going to be here with you up to the end. Thank you so much.
Thank you so much, Dr. Mpamizo, for those kind words. And now I think we can delve into the question and answer session. Dr. Katama, I hope you are listening in. So Mr. Kansimeluk asked the question that, do we perform routine laboratory investigations in Uganda for serotonin and dopamine levels in management of depression?
Well, thank you. for that question. That was from, is it Kansime?
Yes, please.
So looking at particularly investigations, of course, now like when I talked about the imaging modalities where we have to do those spectroscopy imaging, the functional imaging, we really do not have these in Uganda currently. But of course, with the advancement in these concepts that now we are discussing things at the level of the synapse, possibly in the nearby future, we shall be having some of these modalities in our setting. Thank
Yeah,
you.
Dr. Katama, yes, I may agree with you. We agree indeed that serotonin, dopamine, and norepinephrine are central to the neurobiology of depression. And but the reality is their central nervous system concentrations cannot be directly measured through routine blood tests. So since these neurotransmitters act mainly within the brain, but you cannot, how can I put this? They act mainly within the brain, but you cannot base on their peripheral concentrations to determine what is happening within the brain. So yes, I agree with you on the point of spectroscopy and all that, but when it comes to blood tests, I think I highly doubt if we have yet reached there. Actually, I found out that we are not doing them in Uganda, and they are not being done around the world. And that is the reason mainly. Peripheral concentrations of these transmitters are not the same as those within the CNS. Then there's a second question for you, Dr. Katama. Let me quickly look it up. It was from Mr. Situma Geoffrey, and he asked that, is there a possibility of receptors being insensitive to serotonin?
All right. Thank you, Geoffrey. So that one, of course, the answer is yes. There are possibilities of serotonin being of the receptors being insensitive to serotonin. And this could be the basis for use of other medications when you're managing, for example, a patient. You find that you're giving your SSRI, which is basically increasing the serotonin levels, but even with the optimal doses, the patient's symptoms are actually not improving. There could be a possibility of insensitivity. Of course, you can find things like downregulation, whereby because of too much serotonin stimulation, there are decreased numbers of the receptors. So even if the serotonin is high, the effect of that serotonin will not actually be seen within the patient. And then things like desensitization, whereby the receptors are present, but they just do not respond to the serotonin, or they respond only weakly. So yes, insensitivity to serotonin is actually possible. Of course, there are other mechanisms, like the genetic variability within the receptors and others.
Thank you, Dr. Katama. I like the way you seem to simplify this. Then, Mr. Herman Weyenga, you are saying that that was a SPAB session. Thank you, Dr. Katama. So your question is it possible for us to have access to the recorded session? And the answer is definitely yes. We are going to provide links from which you can access some of these recorded sessions in the future as well. Then Zeima Emmanuel, you are saying this is a nice presentation. At least I have learned nutrition involvement in depression. And yes, certainly. Emmanuel, we are what we eat. Certainly. Most of the precursors for most of these neurotransmitters are obtained from what we eat. So I'm glad that this session has been able to add value to you. Then Mr. Baraka Kenja is saying, I have a question on the gut-brain axis, as I would love to know the causal relationship therein, and whether the alteration in gut microbiota is a result of depression and the decreased nutritional intake expected thereof, or whether that alteration is the cause of depression. Dr. Katama. Interesting question.
Thank you for that question. So when you look at the gut microbiota, so within the things that we talked about in this, we have the prebiotics and then the probiotics. Usually, these microbiota release certain inflammatory, they modulate inflammatory pathways. So they release certain chemicals which can directly go into the system and reach the brain, or through the short chain fatty acids, they can also lead to there can be release of the short chain fatty acids, which can be seen to modulate the inflammation within the system. So you're going to find that these microbiota basically is doing a lot in terms of affecting or modulating what is going out of the GIT in terms of either the inflammation or the direct release of chemicals that can actually reach the brain. So it is not just one particular mechanism that is being looked at in this case, but rather a play between different mechanisms that could potentially lead to this depression.
Thank you, Dr. Katama. There is a statement that I like a lot, that there is a bi-directional relationship. And I think you'd agree with me on that. I'm seeing Dr. Emmanuel. Dr. Mwesiga, please unmute and speak. I'm seeing your hand up. Dr. Mwesiga.
Okay, thank you. You've said what I was just going to say. But it's very important to know that the gut microbiome is a very strong regulator of neurotransmitters, brain neurotransmitter, and then the reverse is true. So you can't have this discussion without understanding that bi-directional relationship. So it's something that's very important to know, and it's one of those key drivers in depression, and that's why you will find that many of these other non-pharmacological methods that Dr. Katama mentioned, diet, exercise, they work through that mechanism. Thank you.
Thank you so much, Dr. Mwesiga, for that. Dr. Katama, in 1 minute, Mariam Kasozi asked us that given the genetic influence and involvement in the production of neurotransmitters and neuroenzymes, is there a possibility of having gene editing as part of treatments, maybe in the future? 1 minute, please.
Well, that is a good question, but just as we see that these genetic conditions have been here, there are even those which are even very obvious to everyone, things like sickle cell, but still developments are just showing coming up with, of course, with the recent advancements in technology. Hopefully in the nearby future, indeed, we might have such solutions for these genetic problems.
Thank you so much, Dr. Katama. Dr. Kauba Bubeka, you have asked an interesting question, but unfortunately we are out of time, and we had purposed that we will be requesting only 1 hour from the participants in this session. So probably if you tune in tomorrow, I think Dr. Katama, you can take a note of this question. Probably we shall start with that and proceed. So tomorrow, still at 7 p.m., we are going to wrap up this 3-day series with the final session on treatment principles of depression. This is where the science is going to meet the art of care. Let us all be there. Thank you for having been with us, and have a lovely night.
Thank you, Dr. Myanga, for modulating.